After we've pre-processed our data with recon_all, we can test hypotheses about differences between groups. For example, are there differences in cortical thickness and volume between cannabis users and controls? To answer this question, we first need to organize our data. Here, I've created a subdirectory called FS inside of the cannabis directory, which stores the FreeSurfer output of all my subjects. To make sure the commands run without errors, you will need to specify subjects_dir as the present working directory, and also copy fsaverage from subjects_dir to the current directory. From the directory containing your subject's data, type cp $R $FreeSurfer home/subjects/fsaverage, and then a dot. Then type setenv subjects_dir in backticks pwd. Also create directories called fsgd and contrasts. We'll be using them later. For group level analyses, we will also need to make a file called the FreeSurfer Group Descriptor File, or FSGD for short. This dataset comes with a file called participants.tsv, which contains the subject IDs, as well as information about participant gender, age, and individual difference scores, such as questionnaires. Make a copy of this file and put it in the FSGD folder. Call it cannabisstudy.tsv. Open it up in Excel. For now, we just want to compare the groups, omitting all of the other variables. Insert one column to the left, and then insert 4 rows. In the first row, first column, type group descriptor file space 1, so that FreeSurfer knows it's an FSGD file. In the second row, type title space cannabis study. For future studies, replace cannabis study with the title of your study. Next, we need to specify our groups, which FreeSurfer calls classes. In the third row, type class HC, and in the fourth row, type class CB. If we had a third group, we would create another row for another class. Now remove all the headers and remove all the columns except for subject ID and group. In the empty cells of the first column, type input. The second column is a list of all the subjects, which in our case have the prefix sub- in front of them. Replace the values in this cell with sub- and then the number. Then, save out the file as a tab-delimited text file. You should be aware that text files saved out of Excel have carriage returns that cannot be read by Unix. Use the tr command to get rid of them and redirect the output to a new file called cannabisstudy.fsgd. Now we need to create contrasts. For example, let's say that we want to see where measures like thickness and volume are greater for controls compared to cannabis users. Go to your contrast file and create a file called hc-cb.mtx. Enter the values 1, space, -1. The order of the classes in our FSGD file is important. If we had entered class CB before class HC, then this contrast would be -1, space, 1. Create another contrast file called cb-hc.mtx and enter the values -1, space, 1. Mathematically, this is identical to the first contrast, but with the sign flipped. We now have everything we need to run our group-level analysis. In the next video, we will set up our model and do cluster correction in order to determine once and for all whether marijuana turns you into a lazy, granola-eating hippie who wants to destroy America, or whether it is a miracle drug that can cure cancer and bring about world peace. Stay tuned. We've now reached the stage of group analysis. For example, does Group A, on average, have more or less cortical thickness than Group B, and where? The following commands will allow us to answer those questions. First, remember the QCache option? That resamples the structural maps for each subject to the FS average template and smooths the maps at different kernels. If you've already included that option during Recon All, then we just have to stack all of our maps together for a group analysis. Here, I'm going to be analyzing cortical thickness and cortical volume, but you can analyze any of the outputs in the surf directory that you like. Our first command is MRI's preproc. This concatenates all of the subject maps that we will use for our group analysis. I've created a script that gives you control of looping over hemispheres, smoothing kernels, and measurements. Here I'm analyzing both the left and right hemisphere, only looking at the 10mm smooth maps, and focusing on volume and thickness. Nested within these loops is the command MRI's preproc. It requires the FSGD file we created in the previous tutorial for a list of subjects, and then loads the smooth files from the QCache command. We tell it that all of the images have been resampled to the FS average template. We then specify which hemispheres we wish to create group maps for, and specify the output file. This script takes the study name as an argument, which is the name of your FSGD file. Here, I've called it cannabis study, so run it with tcsh run MRI's preproc.sh cannabis study. The output is an MGH file, one per hemisphere and per structural measurement, along with a log file for debugging. Next, we need to set up our model and run our contrasts. Again, I've created a script which will automate this for the hemispheres and measurements you wish to look at. The command MRI glm fit takes the concatenated image outputs from MRI's preproc and then runs a model at each vertex. The FSGD file specifies which class corresponds to which subject, and the contrast files make contrast maps and calculate the significance of the contrast at each vertex. Here, we've only specified 2 contrast files, but you can make as many as you like. Lastly, the surf command means to run the analyses on the surface of the FS average template. Cortex means to only run the model and contrasts on the cortex, and then we output the contrast files. Run this command by typing tcsh runglms.sh cannabis study. This creates one output directory per hemisphere per measurement. Navigate to the directory lhvolume cannabis study 10.glmdir. There are 2 subdirectories, one for each contrast we specified. The y.fsgd contains our FSGD file. Beta.mgh contains the parameter estimates from the model. See if you can figure out what the other files contain. Within the contrast directories, we have MGH files, which are statistical maps. The sig.mgh file contains the significance at each vertex, while the z.mgh file contains transformed z-scores. pcc.mgh contains correlation coefficients, and gamma.mgh contains effect sizes. We can see the significance maps on the FS average overlay by typing the following: freeview-f, subjects dir, FS average, /surf, /LH inflated, and then a colon to specify the overlay. This overlays the sig.mgh file on the FS average inflated template. Make sure to load the hemisphere that the directory is based on, in this case, the left hemisphere. Also be aware that FreeSurfer uses negative log 10 notation. In other words, a value of 3 corresponds to a p-value of 0.001. Click on Configure to change the threshold of what is displayed on the brain. We're almost there. In the next video, we will cover cluster correction to determine once and for all where marijuana is located in the brain. Finally, we come to cluster correction. In the last video, we used MRI GLM fit to test the significance of parameter and contrast estimates at each vertex. However, we need to correct for the number of tests that we do. One of the most popular correction methods is cluster correction, which calculates the significance of clusters based on the amount of smoothing and how many individually significant vertices are contiguous. This script is similar to the others in its loop structure. The options for MRI GLM Fit Sim are to specify the directory that contains your contrasts. Cluster correction will then be run for each contrast in that directory. The cache option specifies the cluster forming threshold, or what the significance level is for each individual vertex, and also whether you are doing a directional or non-directional test. Remember that FreeSurfer uses negative log 10 notation. In other words, 1.3 corresponds to a cluster forming threshold of 0.05 per vertex. These are cached values for the cluster simulations, which means they have already been run. Your options for the direction of the test are pos for positive, neg for negative, and abs for absolute, or a two-tailed test. Lastly, CWP specifies the cluster-wise threshold, or the significance threshold for the clusters. For most purposes, 0.05 is fine. 2 spaces means to correct for doing tests on both the left and right hemispheres. If you are only interested in one hemisphere, you can omit this. Run this script with tcsh run clussims cannabis study. If it runs without error, you will see new files created in each GLM directory. For example, go to the directory lh volume cannabis study 10.glmdir hc-cb. The names of the files contain the cluster forming threshold and the sign of the test that you did. The most important files are 1. The summary file, which is a text file listing the size and locations of the significant clusters, and 2. The MGH file, which can be overlaid on the FSAverage template. You can use these files to make figures and tables of your significant clusters. Next, we're going to wrap up with correlation analyses and region of interest analyses. In addition to group analyses, you may want to run correlation analyses. This allows you to test whether a structural measurement, like thickness or volume, correlates with an individual difference measure. Remember that our dataset comes with a file called participants.tsv, which contains several potential covariates. For example, we have a measure called audit, which is a questionnaire about alcohol use. In addition, we may want to include age as a covariate, either to see whether thickness or volume correlates with it, or to regress it out of our model when looking at group comparisons. Before going on, let's review FreeSurfer's options for modeling: different onset, different slope, or DODS, and different onset, same slope, or DOS. DODS is the default. It estimates a separate mean, or intercept, for each group in our model, as well as estimating a separate slope for each additional regressor or covariate. DOS estimates a separate intercept for each group, but estimates only one slope per regressor, since it assumes that the slope for that regressor is identical across groups. So, which one should you use? It's up to you. DOS will increase your degrees of freedom and therefore increase your power, since there are fewer parameters to estimate. However, if you think that DODS is a more accurate model of your data, then you should use that. Now let's see how including additional covariates affects our FSGD file and our contrasts. Open up the participants.tsv file and the cannabisstudy.fsgd file in Excel. From the participants.tsv file, copy the age at baseline and audit baseline columns to the FSGD file, making sure they match up with their corresponding subject. Then, add another row below the classes and type variables. Anything written after this specifies the covariates that are in your model after the class column, so make sure they also match the order of your covariates. In the cells to the right of variables, type age and audit. Then save it out as a text file, calling it cannabis study _age _audit, and run the same tr command as before to get rid of the carriage returns. Now go to the contrast directory and make 3 new contrasts. Use the echo command to insert a vector 1, -1, 0, 0, 0, 0 into a file called hc-cb _age _audit.mtx. This will contrast the structural measurements of the groups while regressing out age and the audit scores. Note that since we're using DODs, we estimate 1 parameter per regressor per group. For example, given that in our FSGD file, we first entered age as a covariate and then audit scores, those 2 numbers are the slope estimates for age, and these 2 numbers are the slope estimates for audit. To break it down further, this number is the slope estimate for age for the HC group, while this number is the slope estimate for age for the CB group. Take a guess at the breakdown of the last 2 numbers. If this were a DOS model, we would have 1 parameter estimate per covariate. In that case, our contrast would look like this: 1, -1, 0, 0. This number would be the contrast weight for age collapsed across group, and this number would be the contrast weight for the audit scores slope collapsed across group. Always make sure that your contrast files and your FSGD files match up. One more thing. The more groups you add, the more regressors you need to add to your contrast files. Let's say we had 3 groups and kept the age and audit covariates. If we did a contrast of group 1 versus group 3, for example, it would look like this: 1, 0, -1, 0, 0, 0, 0, 0, 0. That's 6 zeros to the right of -1. The first 3 columns code for group, the next 3 code for age, and the last 3 code for the audit scores. The same breakdown of individual numbers follows the same pattern as in the previous example. Take a guess what the contrast file would look like for DOS. Going back to our first example, let's make one more contrast for the average slope of the audit scores: 0, 0, 0, 0, 0.5, 0.5. Call it auditslope.mtx. Now make some edits to the run GLM script to run these contrasts. Rename the contrasts, and if you want to switch to DOS, specify that here. Then rerun the MRI's preproc script, the run GLM script, and the run clussim script, just like we did before. I've made a higher level script which will run all of them with one command. Make sure to use the name of your updated FSGD file as the argument. When it's done, looking at the group maps is the same as what we did in the previous group analysis video. For example, in the audit slope directory, we can see where there's a significant correlation between audit scores and volume. Next up is ROI analysis. Almost there. So far in FreeSurfer, we've covered group difference analyses and correlation analyses. A third method of analysis is region of interest, or ROI analysis. Recon all parcelates and segments the brain into different cortical and subcortical structures based on 2 separate atlases, the Desikan-Kiliani atlas and the Destrio atlas. The parcellations can be found in the annotation files, which paint each brain region with a different color. Structural measurements have already been calculated in each of these atlas regions and then stored in text files in the stats directory for each subject. For example, the desiccant-Kiliani parcellation statistics for the left hemisphere are in the lh.aparc.stats file. The segmentation statistics across both hemispheres are in the aseg.stats file. To organize these data for input into a spreadsheet or statistical package, we will use the commands aseg-stats-to-table and aparc-stats-to-table. Let's begin with aseg-stats-to-table. You need to specify the subjects, as well as which measurement you want to extract. For subcortical segmentations, volume is usually the only one you will want to look at. You then give it the stats file you wish to extract from and an output file. The Common Segs option will run the command even if every subject doesn't have all of the segmentations. This is useful if there are one or two subjects who did not have a particular segmentation, and if you don't care about that segmentation. You can open up the resulting spreadsheet and organize it however you want. We use a similar approach with the aparc-stats-to-table command for the parcellations. The inputs are a list of subjects, the hemisphere to extract from, which parcellation atlas you wish to use, and a name for the output file. The measurement extraction options are thickness, volume, area, and curvature. You can then organize the resulting spreadsheet to run statistics as you like.